What if one day you take a water sample from your aeration tank and observe it under microscope and you see that the bacteria in your plant are not moving? What does that mean? Does that mean that they are dead? Let me ask you another question. When you think of bacteria, what image comes to your mind? Look at these four images. Which of this is a bacteria? Please pause this video and answer the question in comments. Come on, let's make it fun and interactive. I'll wait. Done? Well, that was a trick question. None of these is a bacteria. Yes, even this image is of a yeast cell. It is not a bacterium. Are these all microorganisms? Yes, they are. But is any of them a bacteria? No, none of them is a bacterium. What are these microbes and what are bacteria? Simply speaking, anything that requires a microscope to be seen is called a microorganism. See, I told you no textbook definitions on this channel, only practical talk. So, bacteria are a type of microorganism. It is very easy to distinguish bacteria from other microbes. They are far simpler in structure and function. All the other microbes that we saw in the picture exist in our ecosystem, in lakes, in ponds, rivers, and the environment all around us. They are critical in maintaining the ecological balance. In fact, without them, life on Earth would cease to exist. It will stop. Some of them may even be present in your sewage treatment plant, STP. But it is very rare to find these kind of microbes in your effluent treatment plant, the ETP. But when we are talking about the secondary biological treatment, we are talking about a very specific kind of microorganism, and that is bacteria. These are the microorganisms that we add to our plant, that we develop, grow, and manage to achieve efficient effluent treatment. This entire series is focused primarily on this one type of microorganism, which is bacteria. So let's take it step by step. How big are bacteria? Well, they are measured in micrometers. Usually, they are 1 to 2 micrometers long. Yes, many are bigger or smaller, but this is just a general size. 1 micrometer is about a thousandth of a millimeter. So, if I take 1 millimeter and divide it into 1,000 parts, one part will be 1 micrometer. That's how good in theory, right? Let's visualize it. Look at this. This is a small pin. Most of us have seen one. Now focus on the tip of this pin. Let us look at the pointed tip under a microscope. The sharp pinpoint is shown in purple here. You can see that the metal has ridges on its surface, and on that surface, you can see those tiny yellow spots. These are the bacteria. Let's zoom in further. Now you can see how incredibly small these bacteria are, even compared to a sharp pointed pin. In this picture, we are looking at a rod shaped bacteria. But that's not the only shape that they come in. Bacteria can be small and spherical, called cocci. They can be spiral shaped, comma shaped, and many more. Some bacteria have flagella. Flagella are like small whip like structures that they use for moving around. These bacteria are motile, meaning they can move. However, not all bacteria have flagella, which means not all bacteria are motile. This doesn't mean that they are dead or inactive. Just like animals that don't have wings are not dead, all animals do not fly. So, bacteria that don't move aren't necessarily dead. They just may not have flagella. There are many bacteria that are non motile, and many critical bacteria in wastewater treatment are non-motile. It is perfectly normal to find non-motile bacteria in your effluent. So if bacteria have flagella to move, do they also have eyes to see where they are going, hands to grab what they find, mouth to eat the food that they have found? No, none of these. No mouth, no eyes, no stomach, no intestine, no legs, no hands, no ears, nothing. Bacteria do not have any body parts like we do. They don't have internal organs or even internal organelles. A bacterium is a single cell, and even within that cell, there are no compartments. There is no nucleus, no Golgi body, no mitochondria, nothing. Without going too much into many biological terms like prokaryotes, eukaryotes, let me explain what a bacteria is in a very simple way. Have you seen these water filled balloons that we throw at each other during Holi? A bacterium is like a balloon, but instead of water, it is filled with a soup of many interesting things like proteins, enzymes, carbohydrates, lipids, minerals, and other macromolecules. Inside this soup, thousands of chemical reactions are happening all at the same time and in the same space. This is something truly fascinating. Imagine a factory where all the ingredients are mixed in a single vessel and hundreds of reactions are happening simultaneously. And yet, these reactions do not interfere with each other. All reactions occur smoothly without any unwanted byproducts under standard temperature and pressure and in the most energy efficient manner. No chemical factory in the world can match the beauty and efficiency with which these bacteria are carrying out these reactions. Believe me, a bacterial cell carries out more number of reactions inside itself than the factory producing the effluent in which it lives. Isn't that fascinating? But how do they do that? And more importantly, how can we use that information to achieve efficient effluent treatment? After all, this channel is not just to admire how wonderful bacteria are. Even though they truly are, our goal is to use this bacteria, these wonderful microbes, to do what we want them to do. How can we manage this microbial factory? In fact, how do we get the right factory to get the results that we want? So, when we say right factory, we are talking about types of bacteria. Let us understand this a bit further. The word bacteria is a very generic term, just like the word animal. All animals are not the same. We have domestic animals, wild animals, animals that live underwater, some that fly, some that crawl, some that climb. They all come in different shapes and sizes, and so do bacteria. They are all just as varied as animals. Just like different animals have different food, different bacteria require different nutrients. They degrade different substrate. And this is where the secret lies, the secret of harnessing the power of bacteria for wastewater treatment. Let us say I have 100 kilos of grass and that I want it to be eaten. Which animal should I get? A goat or a lion? It's obvious. The goat will eat the grass, not the lion. No matter how many lions you get, they will never eat grass. Similarly, if I have a substrate that cannot be degraded by a particular kind of bacterium, no matter what the bacterial count is, the substrate will remain undegraded. The COD will not go down, even with high MLSS or by adding a product with high bacterial count. Bacterial count does not matter if bacteria is not right. We have to use the right bacteria for the right substrate. A goat will eat grass and a lion will eat meat. You cannot switch one for the other. You have to know your substrate and you have to know your bacteria. And then you have to select the right one. And yes, we'll see how to do that the easy way. In the upcoming videos, from video 4 onwards, we'll take a look at how bacteria take in the food. How do they degrade it? and why do they degrade it? What is really meant by bacterial growth and so on. Now I know you might be thinking, why haven't we started talking about BOD, COD or MLSS yet? Hold on, we'll get there. The wastewater system always keeps throwing new challenges and turns in your way. I do not want to show you how to make just one turn. Instead, I want to show you how your car works so that once you know how to drive it, you can navigate around any obstacle, any turn that comes in your way. So, initially, let's focus on your vehicle and the road will take care of itself. When you observe bacteria under microscope, you now know that the movement alone does not define whether they are alive or active. Look at other indicators of activity like cell density, flock structure, and so on. We are going to study flocks in our video number 7. And yes, maintain a logbook, a journal, where you will monitor everything that is happening in your plant. This logbook will teach you a lot. Log everything: the COD, the BOD, the MLSS, the color of effluent, the pH, and the changes in pH, the temperature if you can. On what day, what stream was let in your plant? What was the pH change that happened? How much alkali or acid was added? And so on. And let all these entries have dates. After a few months, you'll automatically start seeing the patterns. Like when a specific stream comes to your system, the pH always does this. 3 or 4 days later, the MLSS goes up or goes down. What happens in winter and what happens in summer? You will be able to predict what is going to happen in your system and be prepared to handle it. It is like having your Google map turned on. The logbook will show you where you are and where you are heading. So maintain a detailed logbook for yourself. I'll see you in the next video where we'll learn about enzymes. Also, there is a link in description where all our videos are organized in one place so that you can watch them without any distractions. I am Sunil Budke and you're watching Evlon Biotech, where we have got bacteria for every problem. Thank you so much for your time. Keep learning, keep growing, and take care.